Hello everyone, I Assistant Professor V Shete from Department of Microbiology, KG Somaya College. In this online lecture, we are going to study the antibiotic resistant and some important factors or the points related to this same topic. So we'll start. Now we will see in this lecture the development of the antibiotic resistance. Then we will study the reasons or the mechanisms which are responsible for developing this antibiotic resistant in the bacteria. Then we will study antibiotic misuse and how we can prevent or how we can take care of this misuse, right, of the antibiotics. Then first, if we will look into the development of the antibiotic resistant, so starting with what is the antibiotic resistant, right? So if we look into the term resistance, it means to resist something, to oppose something. And antibiotic resistance, which means oppose the effect, to resist the effect of the antibiotic on the itself. Now this term antibiotic resistant is the emerging, and it is always the subject that comes into the lamplight for the researchers, because this is the very emerging study. Now there are a lot of bacteria or the pathogens, we can say, that are rapidly developing this antibiotic resistance, and due to which the drugs that we make against such pathogens, then that will be no more applicable to such pathogens, and so that the infection range and the prevalence is getting increased due to this antibiotic resistance. Now if you look at into the picture, there is a normal bacteria. Now say it is not resistant, it is a normal pathogen in its native state. And now if we applied the antibiotic treatment or the antibiotic dosage into it, then what it will do, it will kill that pathogen or it will kill the bacteria. This is the normal scenario. Now if we look into the second below diagram, now it is the mutant cell. What is a mutant? There is a certain genetic change in its DNA, right? Some addition of the gene or some removal of the genes, due to which now the bacteria has developed the resistance against that specific antibiotic. Okay, now it is called as the mutant cell. And so if we now applied the same antibiotic into that mutant cell, now it will resist that effect, and therefore it will continue to infect, it will continue to multiply, right? That we called as the antibiotic resistance, that will continue to reproduce and produce offsprings that resist the antibiotics used to treat. Okay. Now we will see the development, how this antibiotic resistance was developed. Okay, and we will see some of the examples related, the major most famous examples of this antibiotic resistant strains. We will look into that also. Now first, if we look into the definition of the antibiotic resistance, so it can be defined as the microorganisms that are not inhibited by usually achievable systematic concentration of an antimicrobial agent with normal dosage schedule, or fall in the minimum inhibitory concentration range of that drug. So it means that the antibiotic resistant organisms or the antibiotic resistant strains, these are the microorganisms, the pathogens, which are now opposing the effect of the antibiotic that previously was successful effect on them, right? But due to this resistant, they got due to its genetic change or any mutation. Now they will oppose the effect of the antibiotic, right? That is called as the antibiotic resistant strains. Now we can calculate also the antibiotic resistance using the formula, the MIC divided by the MCC. Okay. Now if we look into the evolution of this resistance or the strains which are first discovered for this resistance properties, okay, so it will include the most important strain called as the Staphylococcus. Now we have the VRSA, then the MRSA strains. Now these are called as the vancomycin resistant Staphylococcus aureus and methicillin resistant Staphylococcus aureus. Okay. Now this S. aureus is one of the most common pathogen of the most common infections that they produce, such as the hospital acquired infections, okay, then community acquired infection. And this treatment of this S. aureus is now become very much complicated due to the development of the resistant properties into that organism. They have developed resistant against many antibiotics, due to which it is difficult for us to develop such antibiotics very fast against this mutant strains of this S. aureus. Okay, now first the S. aureus was treated with the penicillin. Then S. aureus developed resistant against that penicillin. Then the methicillin was implicated for the treatment. Again the S. aureus was developed resistant against the methicillin also. And then the vancomycin come into the picture, a newly discovered drug. Then S. aureus was so capable for producing such mutant strain, it got mutated again and got developed resistance against the vancomycin also. Okay. Now the vancomycin is the drug of choice for the therapy of infection due to the MRSA. And as I mentioned, but increase in the vancomycin use has led to the emergence of two types of glycopeptide resistant S. aureus, right? Then S. aureus also developed resistance against the vancomycin too. And therefore, it is very much difficult to treat the infections caused by such MRSA strains or the VRSA strains. Okay. Now, why this resistance happened in the bacteria or in the pathogen, that we will look. Okay, so first origin of the resistant genes could be due to a natural process. Now if we look into the resistant, there are two types of the resistance. First is the innate resistance and second is the acquired resistance. What is innate? Innate means the inside. So the resistant genes which are already present into some bacteria or the pathogen, so that bacteria or that pathogens are naturally resistant to that strains. Okay, they have not mutated or anything, their genetic structure has got nothing changed, but they are originally like that only. Their originally gene structure is such that they are resistant to some of the antibiotics or some of the strains. So that properties we called as the innate resistant properties of the bacteria or of the pathogen. And now if we look into the acquired immunity, we called as the acquired immunity, right, in the humans. That means if we take the drugs from outside, then we developed immunity against that infection. That is called as the acquired immunity. And in similar way, the acquired resistance means what? The bacteria, now we are in concern with the bacteria, so we will look into that example. So now say if a bacteria originally has not resistant to antibiotic X, okay, but we have some other type of the bacteria, okay, which is originally resistant to that antibiotic X. And if this two pathogen bacteria, previous initial bacteria and the mutant or the resistant bacteria got replicated together, then what they will do, they will transfer its genetic material to each other, and then the mutant gene, if it is transferred, it got transferred to the previous non-resistant bacterial strain, then that strain will also be able to develop the resistant against that antibiotic X. So this is called as the acquired resistant, right? Because it got acquired from something outside, somewhere outside. It was not present prior in that original bacteria or the pathogen. This type of the resistant we called as the acquired resistance. Okay, now we may get the question like, why the resistance genes are present or where they come from. So the resistant genes are maintained in nature because of the presence of antibiotic producing bacteria are present, right? We know that we can produce antibiotics from the bacterial strains, right? For example, penicillin we produced from that organism only, Penicillium notatum. So therefore we have in nature the resistant genes, and due to this genes presence in the nature, that can get transferred to other bacteria, to the other organisms, and therefore they will also develop this resistance. Okay, then these antibiotics can act on other bacterial species other than the producer bacteria, right? So as I mentioned, these antibiotics act on other bacterial species, okay, other than the producer bacteria, right? So if we producing the any antibiotic from the bacteria X, so that antibiotic will not be effective on that bacteria, that is bacterial X. It will be affected on the other types of the bacteria except that, right? Then there has to be a mechanism of protection in the host bacteria against the antibiotic that it produces. So there must be any mechanism present in the host bacteria which is able to protect themselves from that antibacterial or the antibiotic effect, right? That is why they are producing the resistance, and that can be the source for the genes encoding resistance. Okay, so these are the reasons or these are the sources from where this resistance is getting evolved. Okay, now we will see this is the evolution or the development of the resistance and the reasons behind that. Okay, now we will see now there is the evolution of drug resistance in the SREs. As I mentioned, there are the MRSA strains, then VRSA strains. Okay, so what are they? First, very initial strain, original strain of the SREs, who doesn't have any mutational genes or any changed genomic structure. Okay, so we will consider that strain. Now the original SREs can be treated, okay, that was able to treat with the antibiotic penicillin, and this was the time at the 1950s. Okay, there at that time the SREs is very well treated with the penicillin antibiotic. After that, the penicillin developed resistance against this antibiotic called as the penicillin. So now we have the penicillin resistant SREs. Then in the 1970s, the methicillin was developed, and another antibiotic which is effective, which was shown the effective results against this penicillin resistant S. aureus. Then after a few years, that penicillin resistant S. aureus again developed the resistant again that methicillin also, that we called as the MRSA strain, methicillin resistant S. aureus. Okay, now after a few years, another antibiotic human developed called as the vancomycin in the 1990s. So now we were having the treatment for the MRSA with the help of the vancomycin antibiotic. Now after that, in the 1997, again MRSA strains developed the resistant against that vancomycin 2. We called as the vancomycin intermediate resistant strains and the vancomycin resistance enterococci. Now there are the two types of the S. aureus was developed against the depending on the resistant properties. First is the VRSA and the VRE, that is the vancomycin intermediate resistant S. aureus, that we will see again in the later slides, and the second one is the vancomycin resistant enterococci. Okay, now that in the 2002 was totally resistant to that vancomycin. See, intermediate means what? It is not completely resistant, okay, but then in the late 2002 it was observed that the MRSA strains was completely resistant to this vancomycin, called as the vancomycin resistant S. aureus VRSA strains. So that's how the S. aureus was developed, so much resistance against so many antibiotics, right? And therefore this antibiotic resistance topic or the study of the antibiotic resistance is always a very interesting and a very emerging subject for the researchers and for the scientists, that they have to develop. Okay, there is a need for development of new antibiotics for such resistant strains. Okay, so this is about the S. aureus. Now you can see in this picture there is the table of the antibiotics. Okay, and again that the resistance was developed in the which year? So the development of resistance to the newly introduced antimicrobial. So you can see if we say the penicillin, then penicillin was approved in the 1943. Okay, and the first reported resistance was observed in the 1940 only. Okay, then the streptomycin was approved by the FDA in the 1947. Resistance was also in the same year. So you can see, we can say that the bacteria has so much ability to resistant or to mutant its genetic structure with very rapidly, right? They are producing resistant to so many antibiotic and in a very short time duration. So there is a constant need for developing new antibiotic against that resistant organisms, right? Now like that we have the different examples of the antimicrobial agents, and against that we can compare the time duration of developing the resistance, right? And if we look clearly, then we can say that there is not much time in between for development of the resistance and the approval of the drug, right? So we can say the resistant is the property which bacteria or which pathogen develops very quickly. Okay, so now we will see the reasons and the mechanism for the antibiotic resistance in the next lecture. In this lecture we have studied the antibiotic resistance. What is antibiotic resistant? Okay, then how it is present and where it is present, then what are the reasons from where this resistance genes can come from. Okay, then we studied these SRES resistant strains. Okay, and then in this table we have saw the different examples of the antibiotic and their resistance. I hope you understood today's online lecture. If you have any doubt regarding this, please ask in the comment section. Thank you for watching.